Hi, my name is Martin Reyes, and I will be presenting my project proposal on the identification of dengue virus serotypes in saliva samples using methods of multiplex PCR and gel electrophoresis. The dengue virus is an arthropod-borne flavivirus that is found predominantly in the tropical and subtropical areas of the world. Due to its transmissibility through mosquitoes, it is highly rampant and a very real threat in these areas. In 2010, over 390 million dengue infections were recorded worldwide, with only about a quarter of these being detectable. The virus is known to cause dengue fever, a type of acute febrile illness which causes the rapid onset of a fever and other symptoms such as chills, headaches, and acute pain. Although not fatal in most cases, some manifestations can be more severe, such as dengue hemorrhagic fever and dengue shock syndrome. These are known to prove fatal if not intervened early in the infection. There are 4 known serotypes that have been genetically sequenced so far, named DNV1, subsequently 2, 3, and 4. They share most of their genetic material, but they are also distinguished with several non-conservative regions to their genome. During an outbreak, one of these serotypes can be found as the dominant strand, or multiple can be found circulating at the same time. Blood samples are collected from patients in affected areas to be tested for the presence of a specific serotype. This is done so researchers may know which variant is most prevalent in a population. Current methods centrifuge these samples to separate the serum from the rest of the blood, as the virus can be readily found here during an infection. Real-time RT-PCR assays are performed on these serum samples to determine which of the primers of the 4 variants bind to the DNA effectively. While the use of blood samples is commonplace for these practices, the moderately invasive nature of collecting blood can be a challenge towards obtaining a great deal of patient samples. However, recent studies have shown that blood-borne pathogens such as dengue can be found in saliva samples in sufficient quantities. Therefore, the implementation of saliva collection in current methods of serotype identification could lead to an easier acquisition of data with little to no compromise in accuracy. Going over some of the pros and cons here, we know that saliva collection has some major benefits, from being overall less invasive and quicker to collect than blood serum. However, it is not without its limits. A traditional limitation with saliva samples is having less viral material prevalent in comparison to blood serum samples. While this could be challenging for performing immunoassays and other varying tests, RT-PCR is highly capable when it comes to working with smaller samples of targeted material. In addition, the use of multiplex RT-PCR over methods allows us to use all serotype primers at once and identify which primers cause its corresponding genetic material to express during the electrophoresis results. Another concern is that the viral prevalence of blood-borne pathogens in saliva is prominent during these acute phase of the infection, but diminishes after this time. This gives blood serum a wider time frame to be taken, limiting salivary samples to the first week of infection. This limitation should be mitigated by the reduction of invasiveness that salivary sample collection benefits from. The goal of this work would be to ease collection of dengue samples by using saliva samples over blood serum. This would be followed by multiplex RT-PCR and gel electrophoresis to confirm results. Over here to the right, you'll see a simplified diagram that conveys the idea of this project. Collection, RNA isolation, cDNA generation, and then a set of primers to be used to identify a specific variant. These would be seen in the electrophoresis results of patients to identify which ones have been found. Identification accuracy should be high considering the promising results of dengue identification in saliva from other published ELISA and PCR trials. Collection of saliva will be performed by having patients spit into a wide-mouthed test tube, which can be stored in a freezer until needed. Separation of the viral RNA from the rest of the saliva will be performed via organic RNA extraction. These can be performed in high quantities with extreme accuracy, but uses toxic chemicals such as phenol and chloroform. Proper care in the laboratory must be taken. The viruses used by Parida et al. To conduct RT-LAMP will be used for designing the RT primers in the study. Non-conservative regions optimal for primers were found by comparing the GenBake sequences with DNA-SIS software and designing the primers with LAMP Primer Design Support software program. While their method of PCR requires more primers, we will only need the forward and backward ones as described by Table 1 here. So you can see in this table, you can see the different serotypes and which target genome they correspond to, and then the primers that were used in the study. Of course, we will only need the forward and backward ones in comparison to theirs, and then the related genetic sequence from 5' to 3'. The creation of cDNA will be through the means of reverse transcriptase, using superscript 3 RT enzyme, which you can see here over to the right, oligonucleotide primers, RT buffer, and treated water. The mixture will be heated in an incubator for about an hour to allow cDNA synthesis and be heated further to terminate the reaction. It will then be chilled and prepped for PCR. We will use a PCR buffer and PFU Ultra 2 fusion hsDNA polymerase, which you can see here to the right as well, with treated water. After several cycles, each taking about 5 to 8 minutes, the PCR products will be incubated for about an hour. Following incubation, the products will be electrophoresed on Argos gel. For this, staining with ethidium bromide allows for visualization on a UV trans illuminator. We will add the ethidium bromide to TBE buffer and run it in a UV box. The results are displayed alongside a ladder, which each strain having specific regions present based on their sizes. Identification of the different strands will be done by comparing the illumination of the gel electrophoresis to known serotype results, such as the one provided right here to the right by Christenberry et al. Through this, serotype of the infected sample will be identified. However, if no genetic material fluoresces, we cannot definitively conclude that the patient does or does not have dengue. It could be that the prevalence of a viral material in the saliva was insufficient. In this case, the timing of saliva collection might be the culprit. It could also be that the multiplex RT-PCR process was incorrectly performed. For this reason, coupling a positive control and a negative control would be beneficial. If continued results remain inconclusive, some improvements to the proposed method should be considered. For once, increasing the number of primers used to detect different regions of the samples could increase result accuracy and reduce inconclusiveness. For example, Christenberry et al. Used over 100 primers of different serotypes in their RT-PCR trials. Different dengue strands and primers than the proposed ones could also be an option in troubleshooting. Regarding concerns about the prevalence of viral material in salivary samples, a dengue pre-screening test can be implemented before working the samples into the multiplex RT-PCR. That way, the presence of dengue can be known well before identifying a serotype. Implementation of this first step would also allow faster identification of infection for those who might be suffering from a more fatal manifestation of dengue. Lastly, safer alternatives to the chemicals used in organic RNA extraction and ethidium bromide staining could be elaborated on. In a setting where the goal is to assimilate as much data as possible, there should be some consideration to the types of accidents that occur with lethal chemicals. That concludes my presentation. Thank you for watching.